Okay, we left off on Monday talking about the role of archaea in digestion in ruminants. And so you remember that in ruminant animals, the carbon source, the energy source that's being absorbed from the rumen is the product of bacterial fermentation, not archaea. So we are talking about archaea in this section. But it's important, and I wanted to make sure people understood this, that the materials such as acetate, propionate, butyrate that are being absorbed here for use in the animal are actually the products of bacterial fermentation. And the role that the archaea are playing is that they're facilitating the continuation of that fermentation by removing the end products of fermentation such as hydrogen gas. Now, again, I realize that this is a module where we're talking about archaea, but since I'm the professor and I have license to do whatever I want to do, I'm going to go off on a tangent for a couple of slides, and you're going to put up with it. Really, this seems like the most logical place to talk about a favorite topic of mine, and that's microbes in the intestinal tract. And how our relationship develops with them. So for the next few slides, forget that we were talking about archaea, although archaea could play into this, and I'll see if I can bring them in. But what I really want to talk about is how it is that we, kind of like ruminant animals, can actually use the end products, and do use the end products of bacterial fermentation as an energy source. So it's not just that we use glucose. We can, very much like a ruminant, use short-chain fatty acids. For the most part, in human health, we're talking about the use of this 4-carbon short-chain fatty acid called butyrate, or butyric acid. This is the end product of fermentation by a lot of bacteria in the intestinal tract, especially members of the genus Clostridium. So we've talked a little bit about Clostridium before. This is very commonly found in the soil bacteria. This is the same genus that has the bacterium that causes botulism. Always confuse anthrax and botulism. But botulism is caused by a toxin produced by a soil bacterium, Clostridium. We've talked a little bit about Clostridium difficile. But most of the Clostridia that live in our intestinal tract, and they are numerically one of the largest groups of organisms in the intestinal tract, are actually mutualists. They're actually helping us in part by producing this butyric acid. So here in this schematized mock-up of the intestinal tract, you'll see that in the lumen shown on the top, you have dietary fiber. So everyone knows you're supposed to eat a high-fiber diet. And high-fiber diets are healthy because they're supposed to reduce colon cancer rates. And for a long time, people didn't know exactly why, and maybe they still don't know exactly why, high-fiber diets are good in terms of reducing colon cancer rate. Many people believed, for the most part, or for many years anyway, that high-fiber diets just added bulk to stool so that the transit time was less. And so cancer-causing chemicals in our diet spent less time in close association with our colon. So the faster food and materials move through the intestinal tract, the less chance of developing cancer. And that's probably somewhat true. But now what we really know is that we actually have bacteria in our intestinal tracts to thank for the fact that, hopefully none of us have colon cancer. But bacteria such as Clostridium butyricum, Butyrivibrio fibrosolvens, these are microbes in our intestinal tract that can do something that we cannot. They can break down cellulose. Everybody knows that we can't break down cellulose. You or I didn't get genes from mom and dad that encode enzymes to break down cellulose. But bacteria do. So now I'll use the air quotes around we. So now I'm not talking about you and I. When I say we, I'm talking about me and my bacteria. So the concept here is the extended genome tenet, or extended genome hypothesis, where what you have to really do when you think about an organism is to think not just about the genetic material that came together at the time of conception from mom and dad. You also have to think about the genetic material that is part of the mitochondrion, which came from mom, not from dad, But you also have to think about the genetic material of the bacteria that you acquired, some of which you got from mom and dad shortly after birth, and probably some of the microbes that you're still contain in your intestinal tracts now probably came from mom and dad. But these microbes, especially members of the Clostridia, are great at fermenting these fibers. So they break down cellulose, something that we can't do, and they produce this short-chain fatty acid, butyric acid. That's actually the energy source for 90% of the energy of the cells that line our colon, the colonocytes. These cells utilize a bacterial fermentation product coming from fiber that you or I by ourselves could not digest. So rather than the extended genome tenet, as I think I have it in the notes here, I prefer a term invented or first coined in 1991 by a hero of mine, Lynn Margulis. She was a famous microbiologist who really came up with the endosymbiosis theory, showing that these mitochondria are actually domesticated bacteria, that they used to be bacteria. So Lynn, and then later on, somebody coined the term instead of holobiont, hologenome, as an idea to consider us as meta-organisms. So what makes so this is actually can get into some pretty metaphysical stuff pretty quick, and we were not going to do that. But if you think about it, it's like, what defines a human? If we're using microbes to digest food products that you and I can't digest, and we're using the products of the bacterial fermentation product, are they not part of us? Are they not another organ? A lot of people, including myself, now have begun to view the human microbiome not as this collection of exogenous bacteria that happens to live within our gut, but as another human organ. So the genetic material, the capabilities of these microbes to produce butyrate, which is used as an energy source, is really a good example of kind of a meta-organism or a holobiont. We are a biological organism which is a collection of not only genes from mom and dad, but genes from bacteria. Scientifically, the way this works is that butyric acid coming from the colon, fermented from fiber, actually is the main energy source for colonocytes because it feeds directly into the mitochondria. So what's being burned in most colonocytes is not glucose coming from the blood system, it's butyric acid. So beta oxidation. Butyric acid is fed directly into the TCA cycle of this mitochondrion. And what that does is that generates all of the reducing power, the NADH, that feeds into the electron transport chain to do oxidative phosphorylation to generate ATP. So 90% of the energy being burned by these colonocytes is coming from these bacteria producing butyric acid. And in a way, again, this is maybe a little bit fanciful to think of, but remember that the mitochondria is a domesticated bacterium. So it used to be a free-living bacteria. So I like to think of this as its free brethren living in the colon are sending in food to their entrapped or enslaved brethren inside the cell, the mitochondria. So there's still communication, in essence, between bacteria, even though one of the bacteria are something that we now call mitochondria. But the other part that's really interesting about this, beyond the fact that these bacteria are producing an energy source, that butyric acid actually has multiple effects on gene expression inside of the cell. So butyric acid can interact, as it's shown over there on the right, with G protein coupled receptors to change gene transcription inside the bacterium. As a matter of fact, most of the genes of the TCA cycle, the Krebs cycle, are upregulated based on signals being received from butyric acid in the environment. So the bacteria outside are turning on gene expression inside the cell. And one of the neat things that happens is that histone deacetylation and histone acetylation is altered based on the amount of this bacterial fermentation product, butyric acid. Now, when this bacterium is undergoing beta oxidation, the mitochondria is burning butyrate to generate ATP. What that does is it keeps the ratio of ATP to AMP relatively high, which prevents the process known as autophagy, so breaking down of the cell. Because this high concentration or high ratio of ATP to AMP keeps turned off this adenosine monophosphate kinase, which would phosphorylate P27, which would promote autophagy. So this beta oxidation is maintaining homeostasis within that cell, and it's also affecting gene transcription based on whether those histone proteins are acetylated. That's actually shown better in this particular slide because this situation in terms of preventing cancer is actually more complex. For instance, butyric acid is known to interact with Tregs, or regulatory T cells. So these are cells that are part of our immune system that play a role in protecting from cancer and protecting from other diseases. But more directly, we know that butyric acid interacting with the mitochondria maintains homeostasis, as I showed you in the previous slides. But what happens when a cell does go rogue? A colon cell becomes cancerous. It becomes neoplastic. One of the things that happens in neoplastic cells, not just colon cancer cells, but a large number of cancer cells, is because they're dividing so rapidly, they switch into what's known as the Warburg metabolism, named after a Nobel Prize-winning physiologist, Otto Warburg. What happens with Warburg physiology is that these rapidly degrading cells now basically kind of try to exist independently of their mitochondria. They're burning much more glucose, and they're burning much less butyric acid. So now butyric acid can rise to higher levels because the mitochondria are largely being shut down. And so butyric acid levels rise. Well, butyric acid levels, when they rise, they actually inhibit the histone deacetylases. So this HDACs. This group of enzymes, which normally takes acetyl groups off of histone, is now inhibited. So you have a shift in the amount of acetylation of histones, which affects gene expression from that bacterial cell. And it promotes apoptosis, programmed cell death. This is what you want to happen in a cancer cell. This is what you hope happens as soon as a cell turns cancerous, before it has a chance to really develop into a tumor. Hopefully, your butyric acid levels will rise. They will inhibit the deacetylation of histone, which will promote apoptosis and decrease cell proliferation. So this is a check mechanism to make sure that we don't develop cancer any more rapidly than we absolutely have to. And it's entirely dependent on these microbes. So rather than thinking of these microbes as just foreign little entities inside of our body, we need to think of them actually as a protective organ, part of our surveillance system against cancer, because they're actually mutualists. They're not commensals, they're actually truly mutualists. Okay, I'm going to get off that soapbox now, and now we're going to go back to archaea, so people can get excited again. Okay, I know maybe you're more excited about the human microbiome. Those of you, before I leave that, those of you who are juniors may be fortunate enough to, in your senior year, take a class that I've teamed up with Bev Scher to teach. So Bev and I taught last year for the first time ever a co-taught senior seminar class on the human microbiome, which was just a fantastic class. Bev, even though she's the hardest working woman in the world, can't do that class every year because she just does too many damned other classes. So we can't do it every year. So this year, she and I aren't teaching together, but it will be offered next year. So those of you who are seniors, you're out of luck for this next semester. But if you're a junior or lower, you can have the possibility of taking I think it's a great class on the human microbiome. And we talk a lot about things like this. As a matter of fact, I stole those slides from papers that we read in the human microbiome last year. But back to archaea. Now, we're going to move out of the we're going to move out of the methanogenic archaea, members of that Euryarchaeota phylum, and we're going to talk about members of another part of the same phylum. But these are microbes, these are archaea that love high salt concentrations. So these are called halophiles, salt-loving members of the archaea. So these are ecologically a relatively restricted group of microbes. There aren't places that have very high salt concentrations in the environment, because these halophiles have a minimum requirement of 9% sodium chloride. So in case 9% sodium chloride doesn't sound very salty to you, just for comparison, our plasma has 0.9% sodium chloride. So if you were making up a physiological saline solution in the laboratory, you would add 0.9 grams per 100 of sodium chloride. That wouldn't taste especially salty to you. Seawater is 3.5% sodium chloride. That's still well below the minimum for halophilic archaea. So our halophilic archaea are not just living and thriving in ocean waters because the salt concentration doesn't even reach their minimum. Most halophiles have a minimum, have an optimum concentration of sodium chloride between 9 and 23%, and they can survive and even thrive pretty well, pretty close to salt saturation. So these kinds of environments, as you can imagine, you don't run across every day. So if any of you have ever, I didn't take this picture, but if any of you have ever flown into like San Francisco or places in California where they make sea salt, what they do is they take sea water and impound it, and then they just let the sun do its work. The water evaporates, and eventually they can collect sea salt from that. Well, in the process of doing that, the salinity gets to be very high levels, and you'll get blooms of these halophilic archaea. That's what you're seeing here. So the color that's coming up in these salt ponds, these high salt concentrations, is actually a pigment that's produced by these halophilic archaea. So you can see it in places like that. And the microbes are really bizarre. The archaea cells themselves are really bizarre shapes. Some of them are even square cells. You don't see very many examples in nature of a biological cell that's square. But they're very odd morphologies. And besides these man-made hypersaline environments, you obviously can go to places like Great Salt Lake or the Dead Sea or some soda lakes. There's a famous one in California that I can't think of, Lake Mono or something like that. But in any case, there's not a lot of them around. But these are places where many times pretty much the only thing that's living in them are these archaeal cells. Now, one of the unfortunate things perhaps about living in a hypersaline environment is that oxygen is not especially soluble in a high salt environments. So oxygen can very frequently be exhausted in these types of environments. These archaea are obligate aerobes. They need oxygen in order to respire. So they use oxygen as their terminal electron acceptors at the end of the electron transport chain, just like you and I do. But they live in an environment where very frequently they're faced with a situation because of that high salt concentration that they can utilize their oxygen use it up very easily. So what they've been able to do is something that I don't think any other organism has been able to do. They've been able to switch from the typical organisms like you and I are, chemoorganotrophic heterotrophs. I know, hear those terms. You thought you were past them for the first exam, but they're coming back again to haunt you. So these organisms are chemoorganotrophic heterotrophs, just like you and I. They do that when oxygen is available, and their physiology is different than ours, but surprisingly not that much different. But when oxygen levels crash, and now respiration is not possible for them, they switch to photoorganotrophic heterotrophs. They switch to being able to use light. This is something that I don't think exists, an example of this exists anywhere else in nature. So these microbes actually have their own classification. They're called mixotrophs. They can mix it up. They can use either light energy or chemical energy. Now, this photoorganotrophic heterotrophs, these are not organisms that do photosynthesis. So trust me, as we go through the next slides, we are not going to go into photosynthesis. I promised you at the beginning of the class, you didn't have to learn photosynthesis, and I'm going to stay true to my word. But what they've done is they've come up with a way to use sunlight to generate proton motive force in a different way. So those bright colors, those purplish colors that I showed you in those saline ponds, those salt ponds, were due to a pigment called bacteriorhodopsin. So this term is a little bit archaic, no pun intended, because it was believed at the time it was discovered that these were actually bacteria. We now know that they are archaea. And to confuse things even further, we have now found examples of bacteriorhodopsin in true bacteria. So this is not something that's exclusively found in archaea. but it was first found in archaea. It's best characterized, I believe, in archaea. But this bacteriorhodopsin is that pigment. So the cells will produce this purple pigment. Oftentimes it's just in patches on their cells. But that pigment, that bacteriorhodopsin, is actually due to a post-translational modification to the protein. So here is the bacteriorhodopsin protein shown in a cartoon spanning the cytoplasmic membrane of this archaea, with the cytoplasm on the inside, the external environment on the outside, on your left. Covalently attached, so this is the post-translational modification, covalently attached to a lysine amino acid inside of a pore through this protein is a molecule known as retinal. It's an unsaturated fatty acid that has, just by as chance would have it, a purplish color. So this is what gives the pigment color. What retinal does is it has the ability to absorb a photon of light. And what it does in the process of absorbing that light at about 570 nanometers, its optimal wavelength, is that one of those double bonds in that fatty acid will isomerize from trans to cis. When it does that, it changes its ability to bind to a proton. So in the trans form, retinal picks up a proton, not exactly from the cytoplasm as this cartoon shows, but from an aspartate just inside the pore. It'll pick up that proton, and then when it changes its conformation because of the absorbance of that photon and isomerizes to a cis form, it donates that proton to another aspartic acid at another location that eventually gets put out of the cell. When that happens, the cis form now reverts to the trans form until it gets hit by another light photon. And you can see that what happens with each photon that hits this retinal, there's the possibility of putting out a proton, generating proton motive force. It's doing the same thing that its respiration chain, its electron transport chain used to do until oxygen was gone. Once oxygen was gone, no way to put electrons across that chain because there's nowhere to put the electrons at the end. So you stop generating proton motive force. This is an alternative way to generate proton motive force. So these protons that are put out here can be used, as we've talked about before, to drive the F0, F1 ATP synthase to generate ATP, so the cell can continue to survive. It can use this proton motive force to drive its flagella so that it can still move. So this is a light-driven proton pump that has evolved in these salt-loving archaea. Here's just another way to look at it, and you can see that, and I'm going to show you an animation in just a minute, which will explain this much better than I can. But here you'll see the alpha helices of bacteriorhodopsin, which multiple times pass through the membrane. So they're forming a pore through the membrane. And so you have aspartates here at position 96, here at 85. The numbers, not important. But these become the proton donors to retinal. So when retinal is in the transformation, it can pick up a proton from aspartate 96. When it gets hit by a light photon and isomerizes one of those double bonds, it will now pass off its proton to aspartate 85. And then aspartate 85 is going to shuttle that proton out. So that's how the proton motive force is generated. So I've got an animation that I stole from the web, and hopefully when I put this on YouTube, nobody's going to shut me down for copyright. So the last lecture, I put up something from the BBC without giving them credit. When I put it on YouTube, it got shut down by the BBC. It was taken off. So that's why I had to put last video back on Blackboard, because the BBC shut us down. I felt pretty important for just a few minutes, like the BBC is paying attention to me. And I think, nope. See if I can make it work this time.
Bacteriorhodopsin is an abundant light-driven proton pump found in the membrane of Helicobacter pylori, a purple archaeon that lives in salt marshes in the San Francisco Bay Area. Bacteriorhodopsin is a multi-pass membrane protein that traverses the plasma membrane of the cell with 7 long alpha helices. The helices surround that is covalently attached to a lysine side chain of the protein. When red now absorbs a photon of light, one of its double bonds isomerizes from a trans to a cis configuration, thus changing the shape of the molecule. The change in red now shape causes conformational rearrangement in the surrounding protein. The light-induced isomerization of retinal is the key event in proton pumping. In the excited state, retinal is positioned so that it can transfer a proton to an aspartate side chain, aspartate 85, that is positioned towards the extracellular side of the protein. Aspartate 85 quickly hands off the proton to the extracellular space via a bucket division of water molecules. The now negatively charged retinal takes up a proton from another aspartate, aspartate 96. This one is positioned towards the cytosolic phase of the protein. Upon reprotonation, the retinal returns to the ground state. Aspartate 96 replenishes its lost proton from the cytosol, and the cycle can repeat. Net result, for each proton absorbed, one proton is pumped out of the cell.
I guess I better let this play. But you can, it's not as efficient, obviously, as an electron transport chain, putting electrons onto oxygen. There's not as many protons going out. But it's better than dying, right? So in the absence of oxygen, they have the ability to persist. Now, I guess that Archaea liked that light-driven proton pump so much, they've adapted it in multiple ways. So they use light-driven pumps in other ways. So here's that bacterial rhodopsin. Here in this cartoon, BR stands for bacterial rhodopsin. So that's the one we were just talking about, where a photon of light hits it, that isomerization occurs, and a proton goes out across it. But there are other related molecules, like halorhodopsin, also found in these salt-loving organisms, that will enhance the proton motive force by importing, using sunlight, to import chloride. Since chloride is negatively charged and protons are positively charged, by importing chloride, that actually accentuates the charge differential across that membrane. So in fact, it accentuates the proton motive force. But again, it's the same situation where you have, I believe it's still retinal, being alternatively isomerized between trans and cis to pump in a chloride being driven by light. They also use it for chemotaxis. Ah, yes, sweet chemotaxis. We always come back to that master, wonderful system. They can use, so these microbes actually can positively chemotax towards red light. So they use sensory rhodopsin 1 to be able to positively chemotax towards light that's in the red range. That tends to be the light that is best suited for photosynthesis. So this allows the bacterium to positively chemotax towards light, but really not for the reason of utilizing that light, so much as trying to get to a site where photosynthesis is going, because they're trying to get to a site where oxygen is present. So they will positively chemotax using this sensory rhodopsin 1 towards positive light. So that's going to interact with a methyl-accepting chemotaxis protein, with our favorite PA, a histidine kinase on the inside. This particular histidine kinase has a way, when it's sensing red light, to keep that bacterium going, so the flagella will continue to rotate counterclockwise, and this little salt-loving archaea will go towards the sun. It'll go towards the light. But it can't go too close to the light. If it goes too close to the light, then it becomes exposed to the damaging effects of solar radiation, blue light, things closer to the ultraviolet range. So the cell wants to not go so close that ultraviolet rays start to damage its DNA or damage its proteins. So it has a completely different sensory rhodopsin, sensory rhodopsin 2, which responds to blue light. Again, it's associated with a methyl-accepting chemotaxis protein and a histidine kinase that can phosphorylate a response regulator that, when it senses ultraviolet light, light in the blue range, it's going to cause the bacterium to tumble. Very similar to the situation that you all know in extraordinary detail already. You have a phosphorylated response regulator, probably something related to, if not the same as key Y, which now will interact with the flagellar basal body to induce tumbling. The bacterium will randomly choose a different direction and hopefully get the hell away from that ultraviolet light before it kills itself. So it finds its way using these sensory rhodopsins, many of which are driven, if not all of them, driven by that cis-trans isomerization of retinal. Okay, so fascinating as that is, let's stay a little bit with hypersaline archaea, but also bring in the fact that there are bacteria that can live in these particular environments. So what are some adaptations that archaea and bacteria that live at high salt concentrations use? So in these high salt concentrations, these microbes have to be able to obtain or attain water balance. Because if the salt concentration is much too high outside of the cell, osmolarity is going to tend to suck water out of it. So these things are going to shrivel up like a raisin and die, right? So what they have to be able to do to maintain water balance is to literally match the concentration of extracellular solutes by their intracellular solutes. Increase this, and what they do is that you can't just put any soluble molecule into the cytoplasm. Certain salts are going to be, toxic. So they put in all of these bacteria archaea to achieve water balance, put in what are known as compatible solutes. So the best examples of these are either amino acids or modifications of amino acids. So bacteria will use things like proline, glycine, or glycine betaine, or betaine, I should say. These are common bacterial compatible solutes. They're compatible because they don't interfere with the biochemistry of the cell. If you just pack them full of chloride ions, that would potentially be dangerous for the bacterium. And so here's just a look at what glycine, for the biochemists in the audience, glycine, the amino acid that would be used in proteins, as opposed to glycine betaine, where you have 3 methyl groups on the nitrogen. So this is obviously an amino acid that's not going to be used to produce protein, but it's non-toxic. So you can increase the concentration of this to match the salt concentration of the environment, so that you don't get the water sucked out of the cell. So halophilic archaea, oftentimes, while this tends to be more of a bacterial situation using glycine betaine, the halophilic archaea tend to use potassium. So they concentrate potassium from the environment, because potassium is actually necessary for many of the enzymes. So these organisms have become so well adapted to a high salt concentration that now their enzymes, more so than any other enzymes, are actually dependent on potassium levels being high. So they use potassium to match the solute concentration in their environment. Now, this is not, so while it might seem kind of arcane to study halophilic archaea, there's actually very clever people that have come up with ways to utilize it. So why is it that you want to understand something about halophilic archaea? We're very glad that this guy did. So this is a very clever researcher, Dr. Das Sharma, at the medical school, University of Maryland. So he's come up with a way to deliver typhoid vaccines. So many areas, so in this country, we don't have to worry too much about typhoid fever. Typhoid fever is caused by a Salmonella typhi, which actually is one of the few salmonellas that spread human to human. Most salmonellas that you might ever get are probably foodborne contaminants, or maybe you got them from putting a turtle in your mouth. Yeah, you never did that. But in many parts of the world, Salmonella typhi, the causative agent of typhoid fever, is still a major cause of death, especially in children. There is a very effective vaccine, but sometimes it's difficult to deliver to certain areas because it can transport can be difficult, storage can be difficult, refrigeration and freezing, consistently being able to get these into some pretty remote areas is pretty difficult. So this guy has come up with a way to put this vaccine inside of a halophilic archaea and have them express it. And so you can just, and since they love salt, and you can dry them down in salt crystals and then just redissolve them, and they're perfectly fine. This is a really clever delivery system that is potentially going to be used to deliver typhoid vaccine to areas of the world where it's still endemic. So this is when I saw this, I thought, okay, this is why it's good that people understand something about these bizarre, salt-loving, archaic microbes, because maybe you can do something clever with something you learn in this class. He never took my class. So, yeah, no. Okay, enough about archaea. So, as I promised in this part of the class, we're now actually talking about microbiology. Not that we haven't been before, but now what we want to do is to go through basically some life histories of bacteria to show you that microbes that might be genetically related to one another, actually quite closely related to one another, can have very distinct life histories, life stories. So now what I want to do is to talk about bacterial diversity among gram-negative bacteria. And so we're not going to worry too much about the fact that there are 17 major phyla of bacteria. We're going to stay within just one phyla, which probably covers many, many of the bacteria that you'll ever know or love. Many of your unknowns, for instance, all of your gram-negatives that you're working with in the lab right now are members of this Proteobacteria phylum. But it's a metabolically and genetically very diverse phylum. Most of the medically important gram-negative bacteria are in here, but also a lot of agriculturally important gram-negative bacteria, as well as many that are very important at helping to recycle elements through the environment. And we'll cover some of that. So again, this is kind of, you almost have to show some kind of a phylogenetic tree when you're going to talk about bacterial diversity. So here it is. What we're going to do is to talk about the global nitrogen cycle. We're going to talk about a bacterial family, Rhizobium, that's capable of nitrogen fixation. So the atmospheric nitrogen that you and I are breathing in and out right now is basically inert. We can't use it biologically. The only organisms on Earth that can take that atmospheric nitrogen and turn it into a form that can be used biologically are bacteria. Bacteria are the only organisms that do this nitrogen fixation. But there are also closely related microbes that are capable of causing neoplastic or cancerous diseases. And so we'll start with the case of cancer, but we'll start with cancer in plants. So I tricked you into taking a botany class after all. We are going to talk about plants and plant diseases. We just won't talk about photosynthesis. So now we want to talk about a disease called crown gall disease, which is one of the rare bacterially induced neoplasms or cancers in nature. Much more common among plants for bacteria to cause cancer, but still not very common. It's less common. There's really only one really good example in human medicine where a bacterium causes cancer. But this bacterium is a bacterium called, here's the name, Agrobacterium tumefaciens. You may have heard of this bacterium before. Some of you may have used it before. You'll see why as we go through this, but Agrobacterium tumefaciens, this cancer-causing bacterium, cancer-causing in plants, is actually the underpinning of all transgenics in plants. So this is how people traditionally, still to this day, move genes into plants, is based on the system that we're going to describe. So the way that this situation normally works in nature is that you have a bacterium living in the soil, very happily, independent of the soil. This is what's referred to as a saprophytic bacterium, meaning that it can utilize just detritus, organic detritus in the soil. But when this bacterium, Agrobacterium tumefaciens, with its Ti plasmid, so this bacterium doesn't have to have this plasmid. There are strains of Agrobacterium tumefaciens that do not have this Ti plasmid, but we're talking about ones that have this plasmid because it's only a bacterium with this Ti plasmid that's capable of causing the crown gall tumor. And if you want an easy way to remember why the Ti plasmid, it stands for tumor inducing. Everything that we're going to talk about in terms of causing the tumor inside of a plant is all on this plasmid, everything. The bacterium by itself is nothing more than a vehicle for this genetic parasite. Again, getting back up onto my soapbox in terms of talking about the true nature of plasmids. They're not a normal part of the bacterial physiology. They are genetic parasites. They are selfish DNA. This plasmid, this TI plasmid, is going to drive this poor little innocent bacterium into being an evil, cancer-causing bacterium. The story doesn't end too badly because this cancer is not actually lethal for the plants. It's more of a horticultural nuisance. You know, horticulturists hate this because they can't sell their plants if they have these crown gall tumors. But what happens is that this bacterium living in the soil can sense a wounded plant in the environment. And it's usually sensing a wounded plant that's very young, a plant that has recently emerged from the soil. Because what happens when a small plant first emerges from the soil, wind or breezes can move that plant back and forth. So minor breaks can occur because the roots aren't moving too much, but the upper part of the plant is. Minor breaks occur in the cuticle or the surface of that plant. So wounds occur. Now, Agrobacterium tumefaciens with this TI plasmid is like a shark that smells blood. They can actually positively chemotax toward this wounded plant because the wounded plant is going to release phenolic compounds. So just as part of the wound and the repair mechanism of the plant, certain materials are released into the soil, which now, because of this TI plasmid, are chemotactically attracting. So the bacterium will move its way like a shark going towards a higher concentration of blood, knowing that there's a wounded animal at the end. Same thing, the bacterium is swimming towards a wounded plant, knowing not only that there's a wounded plant, and the reason why it's important that it's a wounded plant is because it knows it has access. It can get in through that wound. This is what the crown galls can look like. They're almost always very near the soil. There are a lot of reasons for galls, right? So you've all seen these in plants and trees, sometimes up high. A lot of times they're caused by insects. So they're not always, so just when you see one of these things, you don't necessarily want to assume that it's caused by Agrobacterium tumefaciens. But if it's down near the surface of the soil surface, there's a good possibility that it's caused by this. They're not lethal, but they're really ugly. I mean, this plant is probably very embarrassed about having that. Okay, so the way that this works, and this is not to scale, so here's Agrobacterium tumefaciens cell next to a plant cell. So it has sensed a wounded plant in the environment by a two-component signal transduction system that I'll show you in the next slide that's encoded on this plasmid. It has swum its way up to that cell and now has the ability to form a conjugation tube. And again, remember the F plasmid and the ability of the F plasmid to sense an F minus cell in the environment, to bind itself to that F minus cell and transfer a copy of the F plasmid. If this seems like a familiar story, you're right. Because what's happening here is basically conjugation. The exact same situation we talked about with conjugative plasmids, the F plasmid. There are genes encoding proteins for the sex pillars on this TI plasmid, which can form a pore. Now conjugation is occurring not between two bacteria, but between a bacterium and a plant cell. So this is the ultimate promiscuous type of conjugation. Now, through that material or through that pore, what's going to be passed is a section, not the whole plasmid, and not a double-stranded section of the plasmid, but a single-stranded section of one portion of this plasmid called the tDNA, T standing for transfer. This is the region of the TI plasmid that is going to be passed across there with the help of some other proteins, all encoded by this TI plasmid. That piece of tDNA gets into the cytoplasm of the plant cell, passes through a nuclear pore complex, and will be inserted into the chromosome of that plant cell, that poor little innocent plant cell. That TI plasmid, there's a lot known about it. So you can actually, I don't know why this is kind of a trivial pursuit thing, but if in the laboratory, if you grow this bacterium in the lab above 28 degrees centigrade, for some reason it loses this plasmid, and then the cells can't cause any of the disease. So here is, in a little bit more detail, what that TI plasmid looks like and what's encoded on it. So here is the T region. This is the region that's going to have a single-stranded copy of itself transferred into the plant cell and inserted into the DNA. The rest of the plasmid isn't. Now, the plasmid's not lost in the bacterial cell upon the transfer of this tDNA because only a single strand is passed. It's a rolling circle type of replication. Again, very similar to the situation that we saw in the F plasmids. A single-stranded section beginning here at the right border and ending at the left border is what's going to be transferred. But then the other strand that remains is the template to resynthesize that T section again. So the plasmid is not truly lost, and it's not a full copy of the plasmid that's transferred. It's just this T region. But look at what's in this, or the genes that are encoded in this. Two of them, the cytokinin and the auxin, those are plant hormone genes. These are genes that, when they're produced, will produce hormones that cause plant cells to divide. That's basically the nature of cancer cell right there, right? Uncontrolled cell division. These genes, once they're inserted randomly into the plant genome, now are not subject to the normal controls of these plant hormones in the plant cell. They're just expressed full time, all the time. And so the cells divide and divide and divide again. Each time they divide, each of the cells now has a copy of this tDNA in it. That's what makes that gall, that crown gall tumor. But what's also in there are genes here called opine. Opines are modified amino acids. These are bizarre amino acids that the plant can't use. Plants can't use opines to make proteins. They can't do anything with them. They're just a complete waste in terms of the plant. But look what's left on the plasmid back in the bacterial cell that started this whole thing. The genes for opine catabolism. So basically now that enslaved plant cell is producing massive amounts of opines, and now there are many more cells all producing those opines that they can't use, but the bacterium can use them just fine, thank you. So they'll catabolize those and use them as their nitrogen source, their carbon source, their energy source. So they're causing the plant to make food for itself. The genes in this virulence region actually have all of the genes necessary for that type 4 secretion system, which is the same type of system that's used in conjugation. So this is just very similar to the F plasmid. And it's this type 4 secretion system that's going to pass over the DNA and proteins into the plant cell. So I have 2 minutes left, and this is a really good slide, and it's too near the end. So we're going to pick this up again on Friday because this is too good a system to try and rush through. I'll see you guys in the lab, if not, then on Friday.